Hello everyone, my name is Yunfeng. In the past 13 years, I have been engaged in IVD technical review at the Center for Medical Device Evaluation, which is belong to National Medical Product Administration. Now I'm working in Clinical Evaluation and Biostatistics Division 2, responsible for the clinical evaluation of IVD products. I'm very honored to introduce China's IVD pre-marketing regulations and technical requirements to you here. Before we start, I want to make it clear that the definition of IVD I mentioned here refers to a device, whether used alone or in combination, intended by the manufacturer for the in vitro examination of specimens derived from the human body solely or principally to provide information for diagnostic, monitoring, or compatibility purposes. This may include reagents, calibrators, control materials. In some countries also include specimen receptacles, software, and related instruments. Consistent with the international common understanding, China's classification management of IVD is a risk-based classification like medical devices. The risk presented by a particular IVD device depends substantially on its intended use, indications for use, and the intended user. Specifically, the classification of an IVD medical device is based on some criteria. Besides considering the intended use and indications, technical and scientific and medical expertise of the intended user should also be considered. In addition, the importance of information for diagnosis and the impact of the results on the individuals and the public are also important aspects of risk assessment. In China, we divide IVDs into three categories, which are consistent with the US FDA. Some jurisdictions divide it into four categories, such as in the EU's new IVDR. Class 1 include IVDs with the lowest risk, where safety and effectiveness of IVDs can be ensured with routine administration. For example, sample dilution, general culture, medium. Class 2 include IVDs with medium risk, where strict control and administration are required to ensure the safety and effectiveness of IVDs, such as reagents for detecting hormones and proteins. Class 3 include IVDs with the highest risk and must be strictly controlled and administered through special measures in respect to safety and effectiveness, such as reagents for detecting pathogens, for tumor diagnosis and companion diagnosis, and for human genetic testing. There is a close relationship between the IVD classification and its conformity assessment. For Class 1 IVDs, filing is required before entering the market. For Class 2 and Class 3 IVDs, registration is required before entering the market. There is specific information about IVD classification on the NMPA website, which can be downloaded if needed. Based on the classification of product, the supervision of domestic products is managed by national, provincial, and municipal level regulatory administrations. CMDE is a national-level technical support organization that undertakes the function of medical device market access registration review. All imported products are supervised by NMPA, and the CMDE conducts filing and registration review. The main responsibilities of CMDE including to conduct the acceptance and technical review of registration application of domestic Class 3 medical devices and imported medical devices, such as filing of imported Class 1 medical device. To participate in the drafting of relevant laws, regulations, and normative documents related to the registration and administration of medical devices, and other work involving research, guidance, and so on. We are composed of 15 departments, 9 of which carry out technical review work. Among them, the Evaluation Department 6 and the Clinical and Biostatistics Department 2 cooperated to carry out the technical review of IVD products. The IVD management system in China is consistent with the international management guidelines, including pre-market and post-market supervision. From the perspective of the risk management, the safety and effectiveness of IVDs on the market can be guaranteed through the supervision of the whole life cycle of products. Here we will focus on the pre-marketing supervision and technical review requirements. IVD manufacturers need to submit a series of materials to support the safety and effectiveness of the products. After the materials pass the technical review, the products can enter the market. IMDRF RPS Working Group has established a table of contents structure for the pre-market applications called In Vitro Diagnostic Device Market Authorization. There are 6 chapters in talk structure, including regional administrative submission context, analytical performance and other evidence, clinical evidence, labeling and promotional material, product QMS information. This is a worldwide harmonized document. On September 30, 2021, NMPA issued the new announcement on the requirements of medical device registration application materials and the format of approval documents for IVD. We use the ERPS system for application and review, and the ERPS system fully displays the requirements of IVD medical device registration regulations in China in the form of RPS talk structure, which is in line with the international general requirements. We know that IVD products are diverse, involving a lot of methodologies, and their intended use is also very complex, mainly including diagnosis, aid to diagnosis, monitoring, etc. Based on the intended use and the methodological detection capabilities, there are quantitative and qualitative regions. Products of different purposes also have different requirements for their clinical performance. To be clinically useful, the results obtained from a given human sample examined by different laboratories or among different IVDs within a single laboratory should be equivalent, regardless of the measurement procedure employed. In addition to the robustness of product quality, the reliability of laboratories and operators, as far as the design of the product is concerned, traceability is a very important factor that affects the equivalence of result. As stated in ISO 17511, equivalent results for human samples can be achieved by establishing metrological traceability of the values assigned to the calibrators for MP to the highest available reference system components for the variant. Based on the importance of traceability and the accuracy of test results in product development and the verification process, we can see that in the IVD product registration material, relevant data need to be submitted. This includes both the traceability data in the product development process and the verification data after the product is finalized. Firstly, manufacturers should submit research information on the enterprise reference materials, including source, composition, positive and negative or quantitative confirmation, etc. Then the manufacturer shall submit detailed data on the traceability process of the calibrators and shall submit information on the assignment and validation of the trueness control materials on all applicable instrument systems. The calibration hierarchies technical documents should be submitted, including a description of the sequence of calibration and the measurement steps, and estimates of uncertainty of assigned values of measurement in RM deployed at the lowest level in the calibrator hierarchy. We accept the calibration hierarchies model based on ISO 17511-2020. For IVD products, the standard section contains two parts of information: the overall application of the standards for the product and the results of the verification of the national reference material. China's national reference material is a series of physical standards for a certain type of testing prepared by the National Institutes for Food and Drug Control, which is used to evaluate the conformity of IVD products. Generally, it includes accuracy, sensitivity, specificity, repeatability, and other performance evaluation reference materials. When the reviewer has performed a technical review of all the information on the product and has concluded that the product meets the basic principles of safety and effectiveness and can be marketed, in addition to a review report, an output document called PTR, Product Technical Requirement, is produced. This document summarizes the main performance indicators of the product, such as sensitivity, specificity, linearity, accuracy, including precision, etc. This document can be used as a basis for post-marketing performance testing and quality control. Like other countries in the world, in China's review system, the standard system is also one of the bases for the review. Indicators in PTR shall not be lower than the compulsory national standards and compulsory industry standards applicable to the product. Once the recommendatory standards are quoted by the registered or filed PTR, they shall be implemented compulsively. In the pre-market review of IVD products, traceability and accuracy are very important and very basic data. However, an IVD product that meets the requirements of clinical use must comply with the basic principles of safety and effectiveness. Technical data generated during IVD product development, validation, and verification shall be submitted. Moreover, it is important that product development data, verification data, and the validation of clinical data are integrated and cannot be treated separately. The conformity assessment process of a product is a process of synthesizing all the evidence to confirm that the product meets the clinical performance requirements. It can be seen that this is a very complex task. Moreover, the complexity of IVD products also increases the challenges to supervision and technical review. As we all know, IVD products tend to be more diverse, and the scope of the clinical samples being used is being wider. Methodologies tend to be more advanced and diversified, and the laboratories require more automated and convenient diagnosis. The development of the industry has led to the emergence of more new products and new markers. So, although we already have released hundreds of CRMs and RMPs, there are still many cases for variants with methodological traceability only to manufacturers' internal arbitrarily defined RMs. In this situation, how to confirm the clinical performance and results equivalence of different products? Here, I would like to share the experience of emergency pre-market and post-market supervision of SARS-CoV-2 test reagents in China. When we started the emergency review, the SARS-CoV-2 virus is totally new. Well, the clinical demand for diagnostic products is urgent, and the development of CRM needs some time. A large number of reagents are needed clinically, so multiple manufacturers are needed. At the same time, the detection of serious infectious diseases requires high sensitivity and high specificity diagnostic products. This is the situation we face. Relatively speaking, the good aspect is that the PCR method is a relatively conventional method for various detection. Product development has a mature path. In order to ensure product quality, we have advanced a series of requirements. CMDE issued the key points of product review to guide the product development immediately. In the product developing process, the manufacturers established their own enterprise reference material. By using digital PCR, the enterprise reference was established and the detection limit was defined. By analytical performance verification and accelerated clinical trial validation, manufacturers initially determined the analytical performance and the clinical performance. The earliest products had a sensitivity of less than 1,000 copies per milliliter. The China National Institutes for Food and Drug Control has established a unified control material, which were derived from clinical samples. All products were tested by the unified control material. 3 batches of each product are tested to evaluate indicators such as sensitivity, specificity, and repeatability, and so on. We have an emergency approval procedure for medical device. Based on this procedure, we approved the first 4 nucleic acid test reagents on the market within 4 days. Today, we have nearly 11,000 PCR laboratories in our country. Laboratory accreditation and personnel training are the guarantee for the reliability and the recurrence of laboratory test results. National and local Center for Clinical Laboratories continue to carry out laboratory accreditation, routine inspection, and personnel training and education. And also, National and local Center for Clinical Laboratories routinely perform external quality assessment. The latest quality assessment report shows that the National Center for Clinical Laboratories evaluated a total of 8,127 PCR laboratories using samples containing the latest Delta mutations and other mutants. And the result is more than 98% of the laboratories passed. Also, after the product approved to the market, CMDE carried on the product improvement instruction continuously. The enterprise continues to optimize the product performance. And through the change registration procedure, the better product was approved to the market. At present, the sensitivity of nucleic acid detection reagents are below 500 copies per milliliter. Finally, because the virus is constantly mutating, all products on the market are required by NMPA to monitor the ability to detect mutant virus through bioinformatics analysis and verification of the plasmid to track and analyze the impact of mutations on detection. Based on this continuous monitoring data, so far, no failure of the detection for mutant viruses has been found on the market. Also, we required the manufacturers to work on post-marketing clinical data analysis, AE collections, and so on. By October 2021, China had approved 64 SARS-CoV-2 related test reagents for the market, including 32 nucleic acid detection products, 29 antibody detection products, and 3 antigen detection products. These high-quality products provide an effective means of detection for epidemic control in China and the world. Finally, as a regulator and a reviewer, I hope that there will be more CRMs and MPs suitable for high-risk products and more qualified reference laboratories in the future. More importantly, it is hoped that enterprises can improve the research ability of traceability and make products better. That's all. Thank you very much.